Up next is comparative anatomy and embryology. So these two are used to show common evolutionary origins or common ancestors. And this helps support evolution by natural selection because even though organisms look very different as, say, adults, they look very similar when they are embryos, early in development. So these four organisms, the shark, the turtle, the chicken, and the human, these are embryo stages of all four. These are all vertebrates, meaning they all have backbones. And if I had shown you these without the labels, my guess is that you would have had a hard time telling me what organism was what. You probably would have been able to guess this one was a fish of some sort, but the rest of them are a little difficult to figure out. And there's a reason for that. It's because we all start developing roughly the same way. It's after we get to a certain point in our development that we start to specialize to look more like our adult version. So if you look, one of the things that we all have in common at this stage are these two structures that we call homologous structures. Homologous, homo meaning same. In this case, homologous structures, homologous meaning that we have the same ancestor. So we have an ancestor in the distant past that had these structures as well. So let's take a look at them. So all of them have these little orange patches called gill pouches. So on us, they would be up around your chin area, just under your chin, on your neck. Now you don't have them now, right? You don't have gill slits or pouches or anything. Ours get transformed into part of our inner ear. The only organism out of these three that actually has any sorts of gills are the sharks, right? All of the rest of us have actually transformed them into other structures, but we all have them during our early development. The other thing that we all four have are tails, right? But which of these organisms have tails as adults? Like tails that you can see. Yeah, only the shark and the turtle are going to have tails as adults. Chickens have like a feathered tail, so you might have said that. So they will have a feathered tail, but they don't have like a tail like you'd see on a shark or a turtle. And we have a tail, but it's a tailbone, right? It's a very compressed, small structure. Humans are born sometimes with tails. They're not functional tails. They don't move or anything, but you can be born with a tail. So this indicates that we have a common ancestor sometime in the past that had gill pouches and a tail. Since we all still have them, but then we change them before we become our adult forms. Continuing with homologous structures, there are homologous structures in adult forms as well. So these four organisms, human, horse, bat, and porpoise, they are all showing off their homologous structure. Now, if I didn't have the picture with the bones showing up like this, would you think that these four structures were homologous, that they were similar to each other in any way? And why not? All right. Yeah, because most of us look at this and go, well, humans use their arms differently than horses. I mean, horses use their legs to walk on, and bats use their wings to fly, and porpoises use their flippers to swim, right? But if we look, all of these use the same pattern in their structure, right? And sometimes it's a little hard to see, mostly because of how the horse is, but I'm going to show you. If you look, we all have a blue humerus bone, 1. So that's the pattern. We got 1. Then 2 green. Now that's where it's hard with the horse. It's actually fused. That's because of them putting their weight on it. But you can see there's 2 fused together here for the bat, 2 for the porpoise. Then yellow, the carpals. You've got a whole group of those on each of us. They're real tiny on the bat, but they're there. and then a whole bunch of purple, right? A whole bunch of metacarpals and phalanges. And this is the pattern that we see with any vertebrate that has limbs. 1, 2, a group, and a group, right? And this demonstrates a common ancestry between all of us because we all have this pattern in our limbs. All right, so let's talk then about some structures that are a little different. So the human appendix serves no function. Why are we all born with one? We all have one because we haven't gotten rid of the directions for making one, right? We don't use it anymore, but one of our ancestors, our ancestors in the past used it for something and we haven't gotten rid of it. It's gotten smaller probably than it was in the past, but we still have the directions to make it. All right. So we know in something like pigs have a structure called a cecum, which is similar to our appendix, and they use it to digest raw meat. They have extra enzymes and so forth in there, and so there's a good chance maybe that's what our appendix was for. We just don't eat raw meat anymore, and so we don't need to use it. So what is the appendix then called? Well, the appendix is called a vestigial structure. So vestigial structures are leftover structures, evolutionary leftovers. These are structures that are still there but aren't used. They're coded for in the genetics and the DNA, so the organism still makes them, but they're usually a smaller version of the original. So this is another one of my favorite examples. I like the appendix, and I've got some other ones that I'll link a video to for you, but this is the vampire bat. And most people think of the vampire bat bites in and then basically, like, I guess has a straw to suck the blood, but what a vampire bat does instead is she uses these front sharp teeth to slice a gash open on the animal that they're drinking from, and then what they do is they sit there and they they lick up the blood, and so they'll sit there and do that. And that's how they eat. However, if we look at the skull of a vampire bat, they have molars. They have back teeth, which, as you can see here, are adapted to grind up food. Think about it. Those are your back teeth. So why do vampire bats have molars? Because they have a completely liquid diet. They never eat anything they need to chew. Yeah. Their ancestors had them. In fact, all other bats have molars because the other bats eat things like insects and fruit and so forth, and so they still need their molars. So the question is, are there any other vestigial structures in humans? And there are. So I'm going to put a link here for a video if you're interested in it. There are several other vestigial structures in humans. You might think of things like wisdom teeth, but there are other ones that are in our bodies that you may not think of. All right, so there are vestigial structures. Vestigial structures are the leftovers. There are homologous structures, which share common ancestry. And then we have what we call convergent evolution. Convergent evolution is basically evolution had the same good idea, but the structures developed from different original structures. They have different ancestors. And so wings are a great example of this. You know, wings are found in bats, birds, and insects. The starting materials are different, but they have the same function. So, You know, I think we can all agree insects and bats do not have the same common ancestor. So this is what we call convergent evolution. They converge on the same function, but they started on very different starting points, very different starting structures. Okay, so the next section is on molecular biology. So these are different things related to our DNA, our genetic code, showing common genetic sequences linking life on this planet. The first part of molecular biology that shows a strong link for the theory of evolution is that all organisms use the same genetic code, meaning that all organisms write their directions. So they write their DNA using the same 4 letters. So they all use A, T, C, and G, the bases for DNA, to write those directions. And they all use those to build the combinations for making amino acids into proteins. So if you think about it, that's pretty amazing that all living organisms on the planet today use the same language to write their directions and build molecules. And we'll talk more about that later in the semester, how that works. So how then can we find out how related organisms are? Well, we do it the same way that you would, you know, figure it out between humans. So we're looking at the DNA as far as how much there is in common. The more distantly you and another individual are related, the more your DNA is going to be different from each other. So for example, if our whole class had their DNA sampled and we compared it, our DNA would all be very different from each other compared to if your immediate family had their DNA sampled and compared, right? Your immediate family, your DNA should be very similar to each other versus our class. And so that's the same idea that we use here with molecular biology. We take the samples of DNA from different organisms and look at specific sections to see how alike they are or how different they are. And the more different they are, the less related they are to each other. So let me show you an example of this. So we compare these DNA sequences for individual genes. So we're not going to compare the entire set of DNA. That's like crazy. That's a ton of information. But instead we'll take one section. So for example, the gene to make hemoglobin. So this is the molecule that carries oxygen in your red blood cells. Other organisms make this as well. And so in hemoglobin, there are 146 amino acids. And so what they did is they compared the DNA sequence that codes for those 146 amino acids to rhesus monkeys. And on the next page, I have a picture of a rhesus monkey if you have no idea what they look like. And so out of those 146, so let me grab my pen. So there's 146 amino acids in that gene set. And so out of that, 138 were the same as the ones in humans. Okay. And if you think about that, that's pretty good, right? That's a lot in common. 138 out of 146. You know, that means that only like, what, 8 of them are different. And so that indicates that we're probably pretty closely related to rhesus monkeys. And so they did that with other organisms. So I'm going to show you now what the data was. So let me move my face. All right, so here are humans, 146. So 138 of those were the same in rhesus monkeys, so there's what they look like. So 8 are different between the two. Then they tested dogs. So we have 114 in common with dogs, 101 in common with the bird they tested, and then only 21 in common with this lamprey fish. I know, it's very cute. And so what this tells us, we can set up these comparisons and start to put organisms in their family trees, basically. And so this tells us that out of these organisms, we're the least related to lampreys, the most related to rhesus monkeys. And then we have what we call molecular clocks. The thing is that DNA is going to mutate at a fairly regular rate. And so this allows us, that's the molecular clock, it allows us to estimate how long it's been since we had a common ancestor based on the number of differences. And so based on this data, it's been 23 million years since we had a common ancestor. That doesn't mean that we came from monkeys. That means we had a common ancestor at some point in the past that we got this information from. 46 million years ago for dogs, 112 million years ago for birds, and 564 million years ago for lampreys. So it's been a long time since we had a common ancestor with this fish. And so the longer two species have been evolving on their own, the more differences they accumulate. And that, I hope, makes sense. That the more that they're in those groups on their own, the more they're going to have differences.